I'll talk about what type of sensors we're developing for gas and liquid applications, and I'm calling them multivariable sensors. So in a minute or so, in a couple of slides, you will understand what it is. So I'm using the conference laptop, so all my slides are a little bit squished, but we can recognize the things in it, all right? So if the faces will be distorted, then I'm sorry. So for a variety of applications, there are quite a lot of different types of analytical tools, analytical instruments that are used in the field, in the lab. And the reason that they're so big, and sometimes you do the measurements by taking the samples first and then having the kits, because the samples that are being analyzed are not simple, okay? They're very complicated, and therefore the conventional sensors don't help to do those measurements at all, okay? And that's why you can see that for demanding applications, you have a shack where you put your analytical instruments. Those things require periodic calibration, and guess what? These are the gas tanks to bring the gases. If the instruments need to be mobile, then you put them on a truck, okay? You are not discussing that because you would like to do the measurements in the right way. Those examples are when you take the sample in the field, for example, oil, you do those measurements maybe next to your asset and so on. So the idea here behind using these instruments is to have the complex sample and to push the responses to different analytes, different gases, components in a liquid, such as maybe contaminants, into the different directions of your instrument response. And these applications, these are several bullets, they show how broad this applicability of this concept is. And these applications range from environmental and homeland security and all kinds of other ones where those samples are very complicated. Okay, so you wish you have some smaller instruments because you would like to put those instruments maybe as wearable devices, okay, or maybe on a drone, or maybe to have them at home, or to have them in every processing plant, but they are too big, they require quite a lot of infrastructure, and therefore you wish you have the sensors that will be as good as those. So that's why we're developing new concepts of sensing, because as I was showing on the previous slide, there are a lot of drivers, and then when we look at the trends or what the customers are asking, they're always asking, give me the better accuracy of your devices that you call sensors, okay? Give me smaller power requirement, give me probably smaller cost of those devices as well. So we're focusing on having the new types of sensing capabilities that will address these needs. So if we have the complex matrix, either chemical or biological, as you saw on the previous slide, these are the big instruments that are doing the job. They're pushing the responses from different components in the mixture in different directions of your multivariable response of your device. Conventional sensors, we are using a lot of those. You will understand why. But for complex environments, they are responding very similarly to different components in a gas mixture. And that's because they were designed not to be exposed to different components, but to be a detector for a major, for example, leak in the air of some kind that you are not expecting to have any other components. So that's why we are developing the sensing structures. We're calling them multivariable sensors that have not a single output like conventional sensors, but several outputs. And those outputs we're devising to be as much as independent as possible. And then we put these sensors into packaging components. You can see examples of a wearable or asset assembled components. So, and also it's important for us to have the right excitation conditions and the signal modulation conditions in those sensors in order to get the right differentiation between the things in our samples. And from microsystem analytics perspective, we're calling this, we're using this term because we are having a feedback into our sensor to understand what are the best excitation conditions and what are the best processing tools that we use. Okay, so we're using this philosophy across a variety of applications here, and I will show you only a couple of examples of those. So one example will be for gas sensing measurements using photonic and RF sensors, and another one will be for liquids. So if we start with the resonant radio frequency sensors, then you can recognize that very often these are the conventional RFID tags or radio frequency identification tags that have been used for many applications at 13.56 megahertz. These are the called HF tags. And the reason that we're using them because these are the inductor capacitor resistor circuits, LCR resonators, and when we put the sensing materials on top of those, they are responding quite well as sensors as well, in addition to being a tag. So we have two main embodiments of our sensors. So one embodiment, as I mentioned, we put a sensing material onto those antennas, and then the properties of the sensing film that are changed because of the gases in our gas phase, they're affecting the LCR properties of our circuit. Okay, and the second embodiment, that will be the example of liquids detection. We are detecting the properties of the liquid itself because this field propagates a little bit into the out of plane of this antenna structure. So I'm showing this simplified equivalent circuit just to emphasize that we have R and C contributions in our resonant response. When we do the measurements of full resonant spectrum, such as real part and imaginary part, then this simplified circuit becomes quite a lot of more complicated or complex because we know from 100 years of existence of these structures, LCR resonators, that on the low frequency portion it will be inductive reactance, on the high frequency portion of the resonance it will be capacitive reactance, and those features they really those features really boost the performance of our sensors in terms of being multivariable sensor response. And I will show you the examples of how that boost is happening, but when we do the multivariate analysis of our sensor response, we are able to convert our multiple responses from that resonator into the I'm calling them scores plots of our principal components analysis as an example. And the more dimensions in that scores plot we have, the more dispersion or ability of our sensor to reject interferences, to do the measurements of multiple gases, and also to do the measurements in mixtures. I see my next slide perfectly. Okay, never mind. Okay. So the next slide shows examples of our sensing films that we put on top, because we don't want to be late for the second.
There's some time.
Yeah. So the slide that you will see in a second, it shows examples of different sensing films. So you saw those examples of the sensing films from a variety of publications, not only from our group, but from many others, because you will see carbon nanotubes, you will see graphene, you will see now some other types of materials, and that's because we are using them to understand what are the best materials for different applications. So in addition to what I mentioned, you also see the functionalized gold nanoparticles, and also we pay attention on how we are putting the sensing materials into our electrodes, because contact resistance and capacitance of those structures is an important aspect of stability and other features of our sensor. So this PowerPoint cartoon you've seen already, but experimentally we have learned how to push the interferences into the plane that is orthogonal to the plane of our response to an analyte that we are interested in. And because we are a part of a sensing community that have been using different types of sensing materials, these and many other ones, since I would guess 1920s, okay? So we have learned that as a community we didn't win the ability to have zero effects from interferences unless we are a dosimeter or non-reversible sensor. So that's why we changed our philosophy in such a way as that we're pushing the responses to a different direction. We are allowing it to happen, but our requirement and design is to push them in the other direction. And when we do that, then you can see we're rejecting interferences 10,000-fold, 2 million-fold, and that's very important aspect. So we also, in addition to discriminating between different kinds of vapors, we also show this example where we have 10 vapors that are closely related. 9 of them are alcohols with increasing length of the linear chain. These are the homogeneous alcohols from same family, and water vapor as interference. And because our sensors are by design reversibly responding, then you can see that in the absence of the gases, the baseline is nice and flat, and then for concentrations for each gas, it gives the spike and then goes back and so forth. So these are the responses to gases from 1 to 10. And these 4 graphs, because they come from individual channels of our individual multivariable sensor. So when we use a pattern recognition, as I mentioned, such as principal components analysis, then we can see that we can easily get to the 3-dimensional response of our individual sensor. So again, the more dimensions you have, the more ability to reject interferences and to do measurements of individual components in mixtures. And by measurements, I'm talking about quantitation. Okay. So the example of a quantitation is shown on this slide, where we're using the support vector machine followed by the principal component regression. These are the 2 tools that we're using, and you can see we're quantifying individual things in binary, ternary, and quaternary mixtures. So during the question session, if you will ask, is it your biggest number, or you can do 10 components? My answer is, for now, it's the biggest number that we try. We have the system that we can mix up to 10 vapors, and this is something that we will be doing next. I also, in my talk, I don't use the words, maybe I will use it once, machine learning, and these are the tools that we use from our toolbox, because they have been known before that terminology comes into a variety of applications. But you can see these are the single sensors that discriminate or quantify different components in complicated mixtures. So we can use these capabilities of sensors not only for selective detection, but also for very sensitive detection. Here we're showing that when we're putting polyaniline on our sensor tag, and you recognize RFID tag, then we detect the changes in the sensor response at a little bit, I would say less than 3 hours after exposing to volatiles from fish. And I need to say that, so you can see the responses of 3 sensors that were coated with the sensing material and controls, and conventional research grade other sensors that are based on colorimetric readouts it takes about 24, 25 hours for that colorimetric sensor to kick in and have the sensor response. So it shows that sensitivity of these things is also quite important feature. So if we are talking about complex vapor mixtures, then a nice available model system was to do the measurements of volatiles of nice aromas from these teas. We generated the headspace with these flavors, and these are the, again, scores plots from those measurements. The reason that there are two of them, because one of the axes here is the fourth principal component. And that shows us that we are even higher than 3 or 2 PCs in our multivariate analysis. So another application, and I'm showing you only the quick summaries, is for industrial or occupational safety. So the reason that I'm holding this conventional wearable monitor, because for our applications, we were taking 3 days of tests, of training, and these are the instruments that are routinely used for the industrial safety. Okay, that's live. They are not very selective, and people are warning that if you want to stay alive, so pay attention to that. They're not very stable. They're so not stable that your ritual in the morning starts with the bumping or calibrating or making sure that that sensor works. And if you didn't do that, then it's not good for your job security. So you do it every morning before you go outside. So these are the routines because of the current state of these technologies. And we understand that it's current state, and we are trying to develop the approaches that will be more stable and more selective. So this is our one of our prototypes that we're developing to be smaller size, smaller power requirements. And when we are doing the measurements of different types of gases with a single sensor, then these are the labels of different vapors. But if I will remove the labels and I will remove the names of those vapors, then it will be a classic response of a non-selective sensor that is in one of those channels of the multi-sensor wearable device. And that's because of the conflicting requirements of reversibility and response to different vapors or gases. So that's why they are so non-selective. So when we do our multivariate analysis on our responses from our structures, then you can see that we push the responses to different vapors in different directions, as an example shown here. Another important aspect is that we are doing the measurements across a broad dynamic range of concentrations. And here we're showing that we are well above lower explosive limit with our sensors, and that helps us in covering different range of measurements down to ppm levels and percent levels as well. So my next example, as I said, for gas sensors, we will have two examples. One example is RF or measurements or sensors that I showed on the previous slides. And another example is photonic sensors or optical sensors. So to generate color or color changes, there are two principles on this planet to do that. So one principle is to have the chemical reaction or a dye or a molecule that has the electronic structure that will change as different gases are affecting that electronic structure. That's the color changes of different pH dyes, bunch of other dyes. So what this girl is holding is a small plastic plaque of Nephion and crystal violet, because this is our science day. We are bringing kids from different schools, and she's breathing on that small patch, and the color is changing because of the humidity effect. Okay, so very nice, very quick response. The other way of generating the color and color changes by physical principles. And iridescence is one of those principles. This is the photo of tropical butterfly that we have seen many times perhaps. And the iridescence comes from the nanostructure of the scales. And when we are learning about iridescence and how the color is formed and how to change the color, then we're starting to make the individual multivariable sensors that are based on photonic principles. So if we take the butterfly and like that and replicate the experiment that was done about a little more than 100 years ago in Cornell University, where you can put different solvents on the butterfly wings to see what is the matching refractive index that removes the iridescence. So I reproduced that experiment just to see that the color change is quite dramatic from being blue iridescence to yellow or green. But when you are exposing to different vapors, those changes do happen, but they're much smaller. Okay? So again, when you take the optical microscope, then you zoom in and you see that the scales are about 100 by 50 microns. When you take the scales, slice them, put them into a transmission electron microscope, then you see these types of tree structures with multi-layer interference structures that are called lamella. These are the horizontal ones. And when the light interacts with these structures, that's the origin of the iridescence. So we have learned that if we're exposing these structures to different vapors and we're looking at differential reflectivity, then we saw very unusual high selectivity of response to different gases or vapors. So that was in 2007. After that, we started to learn the origin and developing the design rules for artificial sensors, and we have learned through our DARPA work, we reported that in 2013, is that in order to have such selectivity, the chemical composition or chemical structure of this Christmas tree or a tree is different in natural structure. So polarity is quite different from tops to bottoms of these trees, and that's the origin of the selective response. We have learned after that how to design these structures not only to operate at room temperature, because we can make them as polymeric ones, but also at higher temperatures. That's our ongoing work with the Department of Energy, and we are using those design rules to make these types of structures. And these days we are having not only single material of this horizontal lamella, but doing more and more different types of materials to enhance that selectivity as well. So if you look at the reflectivity of conventional butterfly structures, then this is the reflectivity pattern that you can see is not boring. It has some structure, okay? And in order to get even more response diversity, our fabricated structures, as you can see, they have more peaks that are helping us for doing the measurements of multiple gases. So in 2007, as I was mentioning, we were surprised, and we reported that to the scientific community, that the different vapors such as water, methanol, and ethanol, they have this different differential reflectance spectra that in the Scor's plot of a PCA models, they had this three-dimensional response with these three arms that are in different directions. So years later, we found the origin of that, and our simulated results, they looked very similar to the experimental results that we obtained years earlier. And with our fabricated nanostructures, we have started to do more and more gas analysis. That's an example of several gases analyzed with these individual sensors. You see that diverse responses of this single structure to different vapors, and from where you guys are, that diversity is not very large. Even if you come closer, sometimes for untrained eye, they're boring similar, but when you use the multivariate unsupervised pattern recognition, it does the job for you, and that's the result of that pattern recognition. So anybody can get this plot when you have the responses as you measure. So we started to do also measurements at high temperature, as you see, and these are examples of quantitation of CO and hydrogen. Here we're using the support vector machine tools as well. So one example of our liquid measurement. So half of the example is doing the measurements of viable, non-viable cells, and another example is doing the measurements of contaminants in lubricating oils. So the reason that we're doing the measurements of viable, non-viable cells, because to have this information during the bioprocess applications is very critical to have the high quality of your final product. And that's why people take samples and do the measurements offline. When you do those offline measurements, then the those live cells or non-viable cells, they're getting stressed, and your ability to do the correct measurements is decreasing. So therefore, if you have the ability to have these measurements in situ, that will be quite nice. So from biology, we know that those biological cells, they have the different dispersion portions of the spectrum, such as alpha and beta. When we model those, we see that we can easily do the measurements of viable cells because the responses are very strong. Non-viable responses are not as strong. So when, in order to do those measurements, that's why we have multi-resonant response of our sensor. And when we are in the resonant mode, then we are much stronger in the response as compared to the conventional impedance. Those two black lines, dotted and solid, these are the same responses from our samples, but in the non-resonant mode compared to the colored ones. So that's an example of doing the monitoring of the bioprocess run with our in situ sensor and also with the offline measurements. For the reason that we're doing the measurements of oil properties, because existing sensors, although they're installed in different assets, cars and so on, they are not as sensitive as you would like them to have, and they're not discriminating between the different components in oil, such as, say, water contamination, in aging, such as a total acid number. And when we compare the responses of our sensor with the conventional ones, like capacitance sensor and tuning fork, the responses are from 20 to 400 times better than conventional ones. And because we are having this ability to get the measurements of multiple parameters, here we're showing the correlation plots and residual errors for doing the measurements of water and aging as well. My last slide shows this one. Shows that we gradiated to test those sensors in the field. These are the different industrial assets, and you saw those examples of different applications of our sensors, and we're trying to be as close as we can to the more conventional instruments. And I'd like to thank for invitation and for looking at the screen when there were no slides.